Transport in plant is the second topic that we look at when we are examining the plant topics. And this topic about transport is closely knitted with the plant nutrition. So before we start the final part about the important and salient points of the topic, examining the outcomes is something that you need to do. To help you with this, the first part of the outcomes of this topic is Cambridge requires you to identify the positions and not only identify but able to explain the xylem vessels, phloem, sieve tube elements as well as the companion cells in sections of a leaf stem using the light microscope, meaning to say it is expecting you to be able to do it in terms of using slides. Alright, so the explanation should come after you identify the vessels, and not only that, you need to be able to relate structural to its own functions of the root hairs, to the surface area, to water and ion uptake. So structure of the root hairs will probably include the shape of it, which you learned before, the composition of it, and basically the narrow protrusion. C. Explaining movement of water molecules between the plant cell, between them and the environment is another one. So there are two kinds. In terms of water potential only, no calculations are required. Outline means you're able to summarize and of course to describe the pathway which the water is transported from the roots to the leaves to the xylem vessels. From the roots to the leaves, through the xylem vessels. Defining terms transpiration and also explain that is the consequence of gaseous exchange in plants. If we need to add on, I would like to also add on that transpiration, will give you another consequence which generates the transpiration pool. So knowing this, we can begin to examine the salient points of this topic quickly, after which I've revised them already. Longitudinal sections of the xylem and the phloem is commonly asked, and to identify them, let's take a look at the easier one first, which is quite in terms of a drawing. And looking at this kind of a structure, it's clearly seen that the one on the left is actually the xylem vessel. And the xylem vessel, most of the time you will not be able to see this here, so it's actually all sealed up, and it's very highly lignified. So one of the features of it is that it's lignified, and sometimes you get to see some layers of the lignin here, alright, and lignin may be also here. Sometimes for older plants you get spirals like this. If not, generally it is a lumen that is very large, and besides the lumen that is very large, basically there is no cross walls. Alright, so relating the function, you will put actually to reduce the resistance to water flow up the xylem. That's in a nutshell. If you look at it carefully, the one on the right, the phloem basically is a little bit more complicated. So to identify it properly, we can look at it in terms of the whole thing here. If you're looking at the whole thing here like this, alright, this is actually a sieve tube. And you will see this green color thing, or sometimes you will see a little bit of denser part of this. Sometimes they call this one here, alright, to draw it here, then that is called the sieve tube elements. The sieve tube elements are useful in carrying of the thin cytoplasmic strands across this thing here, which is actually the sieve plate. The sieve plate is basically the one which has a lot of holes through it, allow the thin cytoplasmic strands to pass through. Next to it, whereby you will need this cell to carry out its function, is the companion cell. Normally you do not see a lot of mitochondria inside, but generally it is involved in the release of energy through increased respiration, due to the increased number of mitochondria. As such, this is the structure, of the phloem. So transposing these two onto the last one, which is a little bit more, I would like to tell you, don't have to worry about this one here because we are not learning it now, so you can leave it this gaps here. So going back to the last one at the other side, we can identify them one by one also. You don't have to actually pay attention to this part here, because likely they actually consist of the cambium cells, which you already know it. And looking at this carefully, it's easier for us to actually look at the xylem first. You can ignore this arrow here, and this is obviously the xylem vessel. Alright, and actually this one you can also ignore it. So xylem can have actually more than one side by side also. It's in a bundle. Okay, and of course the right hand side right here, when we look at the cell here, that it looks of a typical shape, is this one called the companion cell. And next to it we have also, like I said, the dense part with dots here is actually the sieve tube elements. Alright, the whole thing here is actually the sieve tube. And if they ask you what is this is obviously the sieved plate. So in doing so, you have actually successfully identified the different parts of this. Okay. Now we look at the pathway taken, some of the key processes that actually take place, and what do we actually need to think of when we're doing the descriptive stuff. Alright, a typical diagram will look like this, but may I just bring to your attention that you need to understand that the absorption of the water molecules and mineral salts always starts from the root hair. In any other circumstances, when you see a protrusion until a very long stalk that looks like this, so this got to be your root hair. Root hair cells are too small to be actually seen on the drawing, and so it can't be a root hair. When you zoom in, then you'll be able to see it. This is actually the root hairs. May I make one correction here? The root hair is on the top. The root hair cell is the one that you can actually see, because I missed it out. It is actually magnified 1000 times. Alright, and so the very first point usually we will talk about is that the root hair cell is surrounded, okay, by water molecules in the soil, or sometimes you can say it's actually surrounded by a soil solution. And after that you have seen this part going, then we will continue to say that, so you compare the water potential difference between the root hair cell and the root hair cell, as well as the soil solution, the water potential number 2. After which you will mention that number 2 again, the water will enter by water molecules, please, will enter by osmosis. So using what I said, you can piece out the whole description of what happens after you enter the root hair cell. So here you probably have a lot higher water potential, am I right? Compared to the neighboring cells in this one, am I right? So once it's done, you will see number 3 will happen. So water molecules will move in by osmosis again to the neighboring cells. Then number 4, we usually talk that it moves from cell to cell by osmosis. Okay, until you can ignore this little part here. It will usually go straight into the xylem. Number 5, you will enter the xylem, and once you go into the xylem, you have to go up. Number 6, and it's pulled by the transpiration pull, which is a force that pulls the water up in the stream, which finally reaches the vein in the leaves. If you want to further describe where the water molecules end up in the leaf cells, like the mesophyll cells of photosynthesis, there will be a separate theory. This is strictly from the soil all the way up to the leaves, and that's it. Transpiration on its own, basically we are referring to the parts here that are important is this. Number 1, like I said, water enters the roots by osmosis, enters as a stream of water up the plant by the transpiration pool. Okay, and so sometimes a little bit of water vapor lost through the cuticle, but most water vapor is lost by the transpiration. And when you mention transpiration, you cannot run away from the stomata. Alright, so basically transpiration, you can define it in this way. To measure transpiration, we have a various of ways to actually do it. The potentiometer works in various number of ways and the setups can be different, but potentiometer basically we use as a device to estimate the transpiration rate, and the transpiration is made under the assumption that all the water absorbed by the plant is being transpired. You can only calculate it if this assumption is there. Alright, so it's a typical assumption that we always make before you use the potentiometer. So basically the various parts of it are actually written here, as you can see. To trap the first bubble that you need, you can actually do it at the end here. Alright, and the bubble always move left side towards the plant. The Vaseline to seal the joint between these two, we have studied it before. Between the twig of the shoot and the rubber shooter, there's Vaseline or oil here. And cutting it under water is to prevent the air molecule from being trapped in the xylem vessel. Means you actually incur loss of volume. And the whole apparatus, usually the assembly is actually is done under water because you can pull out certain parts of it. So water reservoir is this famous one here. You can open the tap to drive the bubble back to its original position. Okay, so typical one looks like this. Okay, so like I said, the capillary tube is like this. So just when asked to estimate the volume, you can take its cross-sectional area. Alright, then you multiply by the length moved by the bubble, distance moved by the bubble, so you will get a total volume. Then finally you divide it by the time taken. Rate implies there is a time taken, and therefore you need to also watch your final units here, so that's important. Okay, so the workings of the potentiometer, I'll leave it to you to digest it. The second next part is about the factors affecting the transpiration. As a quick note to go, humidity and wind and air movement are very closely related. To study them together is usually the way to go. And so let's go. First, outside the leaf, let's say you have a certain concentration of water potential like this. On a very humid day, it is actually more than that. We got a lot of them here. So imagine you can see through the leaf in the intercellular air spaces. There's also actually, not so much concentration of the water vapor like this. Comparing the two, you will know that if the water inside your leaf wants to escape by transpiration, it is unable to do so because your air humidity is actually very high. And so then let's conclude that increased the humidity, this humidity is referring to the space outside the stomata, you will decrease the rate of transpiration. Okay, and the conversely is also true. Decrease humidity, increase the rate of respiration. For wind and air, it's a little bit different. When it comes to wind and air, we are talking about the gust of wind coming, and what happens if the wind comes, they may blow away some of your water vapor outside the leaf until it looks like this. Comparing things this way, you can tell that outside now, the concentration of the water vapor is much lower compared to the one inside the intercellular airspace. The water vapor can move out of the leaf by diffusion. Hence, we say that increased air movement or increased availability of wind, you will increase the rate of transpiration, and thus the conversely is also true. Temperature actually affects a very important factor. Any factor which affects or increase the rate of evaporation of water will also increase the rate of transpiration. So now it's very clear already that you know, and another increase temperature on a hot day will increase the rate of transpiration. And of course, the converse is also true. Alright, so to summarize, the three things that we actually look here is this, as well as this one. Alright, before we go on, we have to look at another part of this is about the effect of light on the stomata is a little bit tricky, so we just want to take a look at it closer. So yep. Okay, so basically on a bright day like this, with a lot of light intensity, when the light has been captured by the chloroplasts inside the gut cells, the gut cells will start to photosynthesize. After photosynthesis, they will produce a lot of glucose, which also lowers the water potential inside the gut cells. Then you will look at this now. The water molecules will enter the gut cells by osmosis. And the gut cells will now be turgid. The turgid gut cells will force, and the curvature of the gut cells when they are turgid will cause the stomata opening. Finally, therefore, the stomatal opening will increase in size. But there is a possible shortfall of this. If you continuously do this, the stomata opening will also cause an increased rate of transpiration. So this is basically kind of a two-edged sword that you need to be careful about. Alright, when it's actually too wide, alright, then the client will have to mitigate between these as well as the light intensity effect. On the darker day, like I said, no light. When there is no light, basically there is no photosynthesis in the gut cells. So like I said, there is no glucose produced, and so the water potential in the gut cells would have decreased, which leads to this. Decrease will lead to the water molecules moving out of the gut cells by osmosis. All the arrows out are basically the one out of it. And what happens is that these cells will actually look what? These two will be plasmolized, not turgid, plasmolized. When they are plasmolized, the stomata opening will get smaller. Please take note that it doesn't close completely. Stomata opening will only get smaller, not completely close. Decrease in size. Okay, so study this carefully. The light intensity as well as your rate of transpiration, like I said, is still located. So like I said, they are related, not located. Decreased rate of transpiration as well. These two are actually related. Only for the factor of the light intensity is actually kind of tricky. So study it carefully. You should be able to write it in words, not just those that have actually done it here. Alright.